Dear students, welcome back to my class. Today we are starting with the sixth semester, and in this semester I am going to take one of the paper that is development biology, embryology, and teratology. So now I am starting with the subtopic development biology. So what does mean by developmental biology? Literally, we can say that it is the study of the processes by which animals as well as plants grow and develop. We know that we all are developed from the fertilized egg that is called a zygote. So this zygote undergoes a lot of um, uh, modifications during each time and finally it becomes a multicellular organism or a embryo. So, what are the different kinds of processes which is taking place when an organism is, when a multicellular organism is generally formed from a diploid zygote? So, that is generally called developmental biology. So, it is the study of the processes by which animals as well as plants grow and develop. Then, what are the historical review of this development biology? How this particular branch of biology just formed. It was Aristotle. We know that Aristotle is the father of zoology, and here also Aristotle made the first developmental study, and his observations or his conclusions are generally included in the two famous books, that is De Generatione Animalium and De Historica Animalium. Okay, then later, after the Aristotle's contribution, later comes the Vulture Coiter, who generally concluded or who invented the development of egg from ovary or embryo, or he says that from the ovary the egg is developing, and from this fertilized egg the embryo is getting developed. So, Vulture Coiter says that the egg is generally developed from the ovary and the embryo is developed from the fertilized egg. Then later comes the Van Graaf. Van Graaf generally says something about or who made the discovery of the follicles. You know that follicles are the structural as well as functional unit of ovary. So Van Gogh who discovered this follicle. Then later comes the Antony van Leeuwenhoek, who generally who discovered the sperm. Who discovered sperm? Antony van Leeuwenhoek. Then later comes the Panter. Panter discovered the primary germ layers in chick. What are the germ layers? What is called germ layers? You may be knowing that you may be heard that ectoderm, endoderm, and mesoderm. These are the three primary layers which is formed from a fertilized egg during the process of development. So, these germ layers are first invented by Panter, and he invented this germ layers in chick. Then a lot of theories have been put forward to explain this developmental processes which is taking place in animals. So let's move to the detail of each theory. The first theory is called the preformation theory. This was put forward by Marcello Malpighi, who was lived in 1628 to 1694. And he says that the gametes contains a preformed embryo. That means actually we know that this embryo is generally developed from the different processes, which is different modification which is taking place for the fertilized egg or zygote. But here he says that a preformed or a miniature formed embryo will be present within the gamete. And what is the peculiarity of this preformed embryo? It is very small, transparent, and folded. When embryo began to develop, these parts are also getting developed. So, according to this theory, the gamete contains or the gamete possesses some kinds of preformed embryos, which are actually the miniature form of the large large embryo or the fully developed embryo. When the development proceeds, the size also increases. Some says that preformed embryo developed inside the egg, while some other believes that it is located in the head of sperm. So, there are two strategies are generally found to explain this preformation theory. One says that this preformed embryo is generally present in the egg. Some says that it is generally present in the sperm head, and the sperm head is generally called as homunculus. So within the sperm head, this preformed embryo is generally positioned, and as the development proceeds, this embryo is also getting the size also increases, and it forms a fully developed organism. Then comes another theory that is called epigenetic theory. It was initially proposed by Caspar Frederick Wolfin, who lived in between 1733 to 1794. And according to this theory, eggs are made up of several granules or globules. During development, they gradually organize into layers, that is called germinal layers. These germinal layers transformed into the body of the embryo by its local thickening. So the progressive development of an organism, which is starting from a simple to more complex organization through the utilization of the building units. So actually, this epigenetic theory says that the eggs are generally composed of several granules or globules. And these granules, during the development, all these globules or granules will be organized into a specific layers. For example, germinal layers, that is ectoderm on the outer layer, mesoderm in the middle layer, and endoderm in the innermost layer. So during development, these three germ layers will be arranged in a particular manner. So from this three germ layers, they get transferred to the body of the embryo by means of local thickening. Different kinds of thickening will be happen to these three important layers, and it develops into the final embryo or final organism. So we can literally say that this is a progressive development of an organism which start from a simple to a more complex organization through the utilization of this building unit. Okay, so this epigenetic theory, which is proposed by Caspar Frederick Wolf, was confirmed by another scientist that is called Carl Ernst von Baer, who is generally known as the father of modern embryology. Okay, so we have seen what is meant by preformed theory and what is meant by epigenetic theory. The former one says that the preformed or a small miniature formed embryo will be positioned either in the sperm head or in the egg itself, and during the development, its size also increases. Epigenetic theory says that the eggs are generally made up of small units called granules or globules, and later these granules and globules undergoes a local thickening form three different layers, and these layers then gets differentiated into the distinct body layers of the embryo, and finally a fully formed organism will be formed. Okay. Then comes another theory that is called germplasm theory. You may be knowing that first what is meant by germplasm theory. This theory was proposed by August Weismann, and according to this germplasm theory, a body of an organism is made up of two different types of cell, that is called a somatic cell and a germ cell. The somatic cells generally forms the bulk of the body, and here the characters are not getting inherited. We know that the diploid cells are called the somatic cells. That is, somatic cells are those cells which is including all the cells except the haploid germ cells. So the somatic cells forms the bulk of our body, and those genes which is present in the somatic cells will not be inherited into the next generation. Then the other cells are called gametic cells. You know that gametic cells are actually the germ cells, either sperms or egg. Here the characters are inheritable, in which these characters will be passed from one generation to another generation. So according to this theory, the germplasm, which is independent from all other cells in the body, that is somatoplasm, which is the essential element of germplasm, and this is the hereditary material which passes from one generation to the another generation. So germplasm theory says that there are two different types of cells are there, somatic cells as well as the germ cell, and the germ cells are actually, or germplasms are independent of from all the rest of the body, that is somatic cell, and the genes which is present in this germ cells or germplasms are generally contains the hereditary material which will be passes from one generation to next generation. Then another law comes that is called a recapitulation theory. This is also called a biogenetic law. It was proposed by Ernst Haeckel and Fritz Muller in 1864. Here, according to this recapitulation theory. Recapitulation, which means the ancestral characters will be reappear in the developmental stage of an individual. That means we know that during the developmental stage of a mammal, we can see that during the development or during the pregnancy period, a lot of developmental forms will be much more similar to their ancestral form. That means some forms are fish-like, some forms are amphibian forms, some forms uh, are having its own reptilian characters, and finally it becomes a mammal. So it says that the embryonic stages of higher animals resembles the adult stage of its ancestor. That is called ontogeny. We can say that the ontogeny recapitulates phylogeny. For example, if you are considering amphibian development, you can say that during the development of an amphibia, there occurs a stage which is very much similar to that of fish. We know that fish is actually an ancestor of amphibians. So the fish is actually an adult stage. So in the case of amphibian development, the amphibia development generally shows the adult stage of its ancestor. That is the adult stage of its ancestors of fish-like forms. So that is the fish-like form. So that is called the recapitulation theory or a biogenetic law. Then comes what is meant by determinate egg and indeterminate egg. Have you heard about determinate and indeterminate egg? In the case of determinate egg, the fate of each blastomeres are already specified or predetermined in certain eggs. Those type of eggs are called determinate egg, and those type of development are called determinate or mosaic development. For example, the case of lower organisms like Protozoa, Prolifera, Platyhelminth, Pseudocoelomith, Schistolomith, like Annelid, Arthropod, and Mollusca, as well as Tunicate, we can say that during the development, we know that zygote develops into different kinds of cells that is called the blastomeres. So the fate of each blastomeres are already predetermined. So if we are removing one of this area, a defective embryo will be formed. So in this manner, the fate of each blastomere is already specified or predetermined. That type of egg is called a determinate egg, and that type of development is called a determinate or a mosaic development. Then indeterminate means just quite opposite that of the determinate egg. Here the fate of blastomeres are not getting predetermined. Here this is called indeterminate egg. Here the removal of the blastomeres will not affect its normal development. This type of development is called indeterminate egg, or this type of development, this type of eggs are called regulative eggs. For example, Coelenterata, echinodermites, as well as most vertebrates. Then comes germ layers and the derivatives. I told you that germ layers are primitive layers, which is very much essential for the development or the organization of the body layers of an organism. Three different germ layers are generally formed just after the gastrulation process of an organism. They are ectoderm, mesoderm, as well as endoderm. So I hope you understood what we have discussed today. And the later part of this germ layers as well as in derivatives and its derivative will be discussed in the next class. If you have any doubt, please do in the comment section.